talked about these many times already. I'm gonna, I'm thinking I'm gonna pass around a sign-out sheet for attendance. So these are called GERCs. They're G-protein coupled receptor kinases. Not to be confused with what you might see as GIRK, which that G has to do with conductance, and the I stands to do with current, that are regulated by small G proteins and phosphorylation. So I get confused. I had a conversation with somebody and he was talking about ion channels and I was talking about these GERCs, and it took about 5 minutes before we realized we weren't talking about the same thing at all. So, so here our situation is that once the G protein through the G protein cycle has dissociated so that the alpha subunits go work on their effectors and the beta gammas work on their effectors, then we get a series of GERCs that are going to phosphorylate, generally in either the C terminal or the third intracellular loop. So there are specific phosphorylation sites along here that tend to be in a patch. So in contrast to pKa or pKc that have a really defined motif that you and a computer can identify as a possible pKa site for phosphorylation, these just tend to be serines and threonines that are peppered throughout, maybe 4 of them, maybe 6 of them, maybe 2 of them, and they get phosphorylated. Now, we have what would be 6 different kinds of, or 7, excuse me, GERCs. And the GERC 1 and GERC 6 are both found in the retina. So they are for rods and cones for signaling. You know, as with everything else we've talked about, the rod outer segments have their own little series of proteins that are involved in signaling from rhodopsin. And so they've got their own individual GERCs. So when we think about nomenclature, if you're just seeing the word GERC and it's got numbers, then 1 and 6 are limited to that tissue. Everything else is pretty ubiquitous in the rest of the body. On the other hand, if you're looking at what would be a beta adrenergic receptor and a beta arrestin, then you go with the rest of the beta nomenclature, in which case GERC2 would be beta for beta arrestin binding. So, so the 2 that I'm going to show you a little bit about today are GERC2 and GERC5. And they've got some interesting regulation. Not to say that the others are not important, but on the other hand, if you're going to try to begin to think about things, it's probably better to think about a narrower group, and then you can understand what some of the differences are a little bit later. So once the beta and the gamma subunits and the alpha have dissociated and left, GERC2 can bind to the beta gamma subunit using a plextrin homology domain. Now, you remember that the plextrin homology domains will recognize lipids, but they also recognize what would be hydrophobic sites on some proteins. So there's a limited number of proteins. They're pretty selective, but they're going to recognize hydrophobic proteins, and they're going to bind. So this binds via the pH domain, and this is the only one of all of those GERCs that actually works this way. So you can see that once your alpha subunit is left and you have your beta gamma tethered here to the membrane, this is a very good site for now the GERC2 to be called to the membrane, bind to the beta gamma subunit, and then it begins phosphorylating on some of these phosphates for serines and threonines. So our selectivity here is largely related to, first of all, we don't know very much about which betas and which gammas, but it tends to be a general purpose kind of binding. And then the GERC can also interact with other proteins so that it can remain here as a scaffold, or it can allow these phosphorylated serines and threonines now to call in the beta-arrestin. So we really have two different kinds of signaling pathways that could occur from just simply having this kinase bound itself, or having it phosphorylate the site and then letting beta-arrestin bind. So our signaling can really be related to a number of other kinds of pathways simply by going through this GERC phosphorylation. So the GERC itself can regulate a protein called AKT, which is a protein kinase, and it can block it, or it can block somewhere along in the ERK pathway, or it can interact with PI3 kinase to regulate phosphorylation of the lipids in the membrane. So it's got lots of sites wherein it can actually begin working itself while it's bound here. So this pretty much lists and compares our GERC2 with the next GERC we're going to look at, which is GERC5. But if we look a little bit here at the domains that are found in the GERCs, we've got a catalytic domain, which constitutes most of the middle. And then we've got the regulation at the N-terminal and the C-terminal domains of the protein. So our N-terminal domain is going to allow us to interact with beta-gamma, but then we've also got some amino acid residues that will interact with the beta-gamma over here in the C-terminal domain. So that's what's going to bind us initially to the membrane so that it can begin phosphorylating. But the other thing that you'll see here is that we've got a site for binding caveolin, which means that this particular kinase can be involved in lipid rafts. And we also see that it's got a binding site for calmodulin. So what does that tell us about this particular GERC? It's sensitive to calcium, yes. So you can have a regulatory mechanism here that can regulate this particular kinase. Now here's our plextrin homology domain here, where it can interact with the beta-gamma subunits. So that's going to be the primary mechanism that calls it to the membrane. We also have our interaction sites for the two kinases, and so this would be what binds us to those kinases so that the GERC catalytic subunit can interact with those. Now we have another one, GERC5, and GERC5 you can see is also regulated by calmodulin, also can interact with the caveolin, which is the primary protein that can bind other proteins in the lipid rafts. And we also have some interactions here with another protein called alpha-actinin, which would imply that we can also interact with the actin network that's directly underneath the membrane. So this I've shown you before with our GERC2 regulation. It can be regulated by other kinds of things. And we just talked about the calcium and the calmodulin, which its regulation is going to be a negative regulation, so that in high calcium levels we're going to see an inhibition of our GERC. We also see that when it's in the lipid rafts, it's not going to be active. And very typically, when caveolin does bind to a protein in the lipid rafts, it may be securing it there, but it may not allow its activity. And then the release from the lipid rafts will allow this to go into the proteins that are in the plasma membrane, non-lipid raft proteins, in order to work there. So the fact that this says that it's inhibitory may tell us that this is sequestering it in the lipid rafts, and the regulatory mechanism would be to release it so that it can come out into the plasma membrane components. Then we have some phosphorylation regulation. We've got ERK, which also can inhibit, and we've got a protein called SARC, which is the non-receptor tyrosine kinase that's in the cytosol, and this is going to be positively regulating by phosphorylating out some tyrosines. Our two serine threonine kinases are going to activate, so this could be very useful if you have a protein interaction through a G-coupled protein that is now just produced a regulation of a GS that created cyclic AMP in the area, or a GQ-coupled receptor that perhaps just brought PKC to the area. So we've got our regulation here that's going to activate. So you can see that you can get a good number of regulations, not necessarily stimulations, but inhibition that can keep this kinase suppressed when it's not being used. Okay, now we want to go a little bit to a new subject altogether. Does anybody have any questions about the GERCs in general? I should oh, yes, Nathaniel. Okay, so the pH domain was that region that is on proteins that can interact with lipids in the membrane, but it can also act on some proteins that have hydrophobic domains that are that it can interact with as well. Because remember, it's a charge-charge interaction, and but it's charge and lipid, so it can interact with the certain hydrophobic proteins. So this just happens to be one case in which the plextrin homology, the pH domain, can interact with the beta-gamma subunit. Okay, now I want to give us a little update on glycogen synthase kinase since the 1960s, when it was originally recognized as being the kinase that regulates glycogen synthesis and breakdown. So you remember we talked about this on day one with respect to what cyclic AMP can do through protein kinase A to initiate a kinase pathway. So just to refresh your memory, this is going to be taking place in livers and in skeletal muscle, where we have a lot of glycogen stored. So we need to be able to respond to, let's say, a flight or fight response when epinephrine is released, or in the liver, more likely it would be glucagon's normal regulation of maintaining blood glucose levels. So either one of those G protein coupled receptors can be involved in allowing our glycogen breakdown in response to a need for blood glucose to get to the brain. So because of that, our pKa initiates a pathway which can break down the glycogen and allow glucose to be released, or can inhibit the production of glycogen so that we don't have two competing activities. So it's a pathway that involves a couple of kinases. pKa, once it's activated by cyclic AMP, phosphorylates a kinase that's a glycogen phosphorylase kinase that is active. It phosphorylates glycogen phosphorylase, and that breaks down the glucose and leaves it as glucose 1-phosphate, where it can be released from liver into the bloodstream. Doesn't generally get released from the skeletal muscle, though, because skeletal muscle likes to keep its own glucose. So, on the other hand, at the same time, it's inhibiting glycogen synthase. So it does it through glycogen synthase kinase, and when the pKa phosphorylates it, it inhibits it. So, so this is a process where instead of the phosphorylation being an activator, here the phosphorylation on this kinase is to inhibit it. So it's now been recognized, and of course it got that name because that was the kinase that we thought was important. There are other kinases that were named because of their substrates, like myelin basic protein kinase. It's used quite a bit as a general purpose substrate for kinases, certain kinases, but that's not really the be-all and the end-all of their substrate specificity. So the same thing is true with glycogen synthase kinase, because we've now discovered in the last 10 years that it's very important in regulating a number of functions. And it's a protein that tends to be regulated and primed by other phosphorylation by other kinases. So the way this works is much the same way that we just discovered with the pKa, where the phosphorylation can regulate this kinase. So our activation is actually by a phosphatase that will take off this phosphate that's at a serine number 9. So while it's got this phosphate on, it's going to be inactive, and the primary regulation, one primary means of regulation, is to simply take this phosphate off, and then we can begin to get our active glycogen synthase kinase. Now, the one that's probably the most well studied is GSK number 3, and in particular 3 beta. But it's got lots of functions in lots of different places. So, so one place would be at the level of microtubules. And here it's interacting with a cyclin-dependent kinase number 5 that can interact so that it phosphorylates its substrate, which is tau, first. And once the tau substrate gets phosphorylated with CDK5, then the glycogen synthase kinase can put a phosphate on it. And it's now been phosphorylated by 2 kinases, and now this becomes an active protein on microtubules. Here we see another protein. This is the glycogen synthase itself. It gets pre-phosphorylated by chem kinase 2, and when that phosphorylates, then it allows glycogen synthase kinase to come in and phosphorylate. And here we have CREB. CREB was that little protein that was activated by pKa in the nucleus for regulation of gene transcription. And once pKa phosphorylates CREB, then glycogen synthase kinase can phosphorylate it as well. So, so we really see that it works hand in hand. It augments any activities that may have worked in advance. So glycogen synthase kinase itself, all these substrates need to be phosphorylated in advance by another kinase before they can be phosphorylated by glycogen synthase kinase B. So here's the way this works. We've got our prime substrate, which has a phosphate on it. In order for it to interact with the glycogen synthase kinase, this phosphate has to come in and close off an auto inactivation site, or a pseudo substrate site. So it comes in, it actually finds its little position here near the catalytic subunit and covers over a site that would have been the inactivation site. And once that happens, then the glycogen synthase kinase can put the second phosphate group on it, and then it leaves that region altogether. If it doesn't have that ability to do that, then what we see is that the glycogen synthase kinase itself can get phosphorylated. The way it's inactive is that it it gets phosphorylated, and then that phosphate group won't allow this new substrate to come on. And it tends to close off part of this, so this would be serine number 9, up here at the near the N-terminal site. So it closes that off, and we call that an autoinhibition. But it's like a pseudo-substrate, so that once we've got a protein that can phosphorylate at this site, and here's an example of the AKT kinase, once it gets phosphorylated here, then it clamps down as if it were the substrate and precludes any substrate binding, so it just can't get into that once it's been closed off. So the first thing to understand is that the glycogen synthase kinase has to have this phosphate removed in order to open up this site so that the primed phosphorylated substrate can put its phosphate here in this region and get phosphorylated a second time. So that's an interesting mechanism, and there are several kinases that are going to be able to inhibit our glycogen synthase kinase by putting this phosphate on the serine 9. AKT, PKC, and PKA will all actually do this. Now here are some of our regulations that can be involved in some of our kinase sites. So we just talked about these proteins being able to be phosphorylated, and they have to be primed by certain kinases first. What we can also see here is this mechanism that we just talked about. And here is what is going to be potentially blocking this. So conceivably, if we had a G protein coupled receptor that was going through the phospholipase beta, so that we activated our PKA, that could inactivate our glycogen synthase kinase. Or we could be going through a PKA mechanism where our cyclic AMP is being produced by a GS coupled receptor, and that PKA could inactivate our kinase. This is the pathway by which our AKT can inhibit. So AKT is one of those proteins that has a plextrin homology domain and can bind to our proteins that have, or our lipids that have just been phosphorylated. And when it does, it is called to the membrane. It binds to phosphatidylinositol 3,4,5 polyphosphate. And then it is the substrate for a lipid-dependent kinase that can then phosphorylate AKT. And when AKT gets phosphorylated, it becomes active. And it can either leave the membrane or stay on the membrane. What can initiate this would be through our insulin receptor or any other receptor that's going to be a growth factor receptor that can interact via its phosphotyrosines with this kinase, which is called PI3 kinase, that we already have talked about, that can start phosphorylating these inositol phosphates. So we can actually get this activated through a growth factor kind of receptor. it simply has to now allow these lipids to become phosphorylated so that they can bring the AKT to the membrane. Okay, so our responses can be long-term responses or short-term responses, and we can get our sustained responses through our beta-arrestin pathway after we've gotten a very short PKA pathway. So one of these mechanisms is through arrestin that can bring the AKT to the membrane. So while we just talked about this, AKT is coming to the membrane via this growth factor receptor mechanism. Another way to do that is through the beta-arrestin. And typically this is going to be occurring for a much longer period of time than simply the PKA activation. So how does this work? Well, after our G protein coupled receptor has already lost its alpha and beta subunits, it's become phosphorylated, the arrestin binds, then the arrestin can serve as a scaffolding protein. And we've talked about several of those pathways already, but you can see that one of the proteins that it can bind is the AKT. So the AKT can interact with this beta-arrestin, where it's going to be also interacting with a phosphatase called protein phosphatase 2A, that can take one of these phosphates off. And then we have what would be an activation-inactivation situation. So when it's got the phosphates on, it's activated. When it takes the phosphates off, it becomes inactive. So this kind of regulation, getting our AKT activated, can now allow what would be a relatively long-term set of changes in the cell that could go on for hours, in contrast to our very short-term regulation by PKA. So this can set up a pathway in which we can get a complex of AKT and inactivate it. So if it's inactivated, it can no longer phosphorylate glycogen synthase kinase. So if it doesn't phosphorylate glycogen synthase kinase, that leaves glycogen synthase kinase as an active protein. So our whole pathway that might go through this pathway, because we can inactivate our AKT, can actually allow a continued activation of glycogen synthase kinase over a period of time. So how can this be useful? Here's an example of a dopamine receptor, serotonin receptor responding to a large number of drugs that can block them or activate them, as the case may be. And so these receptors can bind to beta-arrestin. So here's our example of our D2 receptor binding to beta-arrestin. That can call in the phosphatase that can take the phosphate off the AKT. Once the AKT is inactivated, that allows glycogen synthase kinase to become active, and it can interact with its target tissues. We could also go through an example here of our serotonin receptor. When these receptors are activated, they could conceivably interact with the glycogen synthase kinase and via their mechanisms of action to allow its activation or its inhibition. GSK3 has some synthetic antagonists that are typically used in the research literature that can block this pathway. I think one of your questions yesterday, James, was how do we recognize some of these things that may be particularly blocked, and these particular inhibitors are very useful here for separating what would be this kind of mechanism through GSK from some of the other mechanisms that would be through the PKA or PKC site alone. Okay, now our phosphorylation by PKA and PKC are going to actually inactivate our GSK, and we just talked a little bit about how that would happen. One of these pathways that this could happen in is regulation of our microtubule associated proteins, and here we've got two of them. One is tau and one is the MAP, microtubule associated protein 1B. So these are proteins that are going to allow the breakdown or the production of new microtubules. So they're rather important, and they're regulated by our glycogen synthase kinase. We also have other protein complexes throughout the cell that are going to be regulated by our glycogen synthase kinase in addition to the microtubule. Here is one that is going to allow proteins to go in and target the regulation of new protein synthesis through beta-catenin. So in this case, we have a pathway that is initiated by a protein receptor called Wnt, and this has a complex of proteins that can interact with glycogen synthase kinase and allow release of the beta-catenin, which is stored within this protein complex in the cytosol. And once it's released, then it can be allowed to get into the nucleus and allow new protein synthesis. And here is some interaction with our Alzheimer's disease, and this is through the protein tau that we just talked about. So our tau protein, once it's phosphorylated with the CDK9, can get phosphorylated by glycogen synthase kinase, and our tau regulation is dysregulated during Alzheimer's disease. So that gives us a little bit of insight into some of the complexity here with glycogen synthase kinase. And I did notice that in the lecture set that we had on our mTOR that we had kinase signaling through this pathway again with So you might go back and take a look to see where glycogen synthase kinase was interacting back with the mTOR pathway as well. Now I've got two phosphatases that I want to talk to you about today. One is a representative serine threonine phosphatase that will take the serines and the threonines off of proteins, and the other one is a tyrosine phosphatase. And again, this is not the be-all and the end-all of tyrosine and serine phosphatases. There are probably about 100 of them, and so we're just going to take a little look at some broad generalizations by just looking at these two. So the first one I want to talk about is PP2B, and that stands for phosphoprotein phosphatase, and it's also known as calcineurin. There's one thing I wanted to mention that I completely forgot when we were talking about AKT, which is a kinase. It's also known as protein kinase B. Protein kinase B was simply given its name because A and C were already used, and they thought protein kinase B would be a reasonably good name, but I don't think it's used very much. So, so the nomenclature AKT tends to be more used. But if you do see something that says protein kinase B, that's what it's referring to as AKT. So here we've got a protein that has two different names as well. So it's also referred to as calcineurin because of its calcium regulation and protein phosphatase 2B. So you'll see it either way, and just to understand that it's exactly the same protein. It's just that in the research literature, it had been named by two different kinds of laboratories studying what they didn't realize was exactly the same thing. So it makes it really difficult for us to follow the research literature sometimes, but typically any group that's going to have a slash in between two names really means that it's got two names and can go by either one. So calcineurin is a heterodimer, which means it's got two subunits, an A and a B. And the calcineurin A is about a 55,000 molecular weight protein, and the calcineurin B is much smaller, about 20,000 molecular weight protein. So they can interact so that we have regulation. Now you notice that this has an autoinhibitory site as well. And this is a very typical way of regulating protein phosphatases and protein kinases, is that to have embedded somewhere in their protein structure something that actually looks a lot like a substrate, so that that substrate can actually cover over the catalytic site and keep the protein inactive until something happens to open up that catalytic lip or catalytic loop. So you'll notice that in all of those diagrams that I showed you from some of the previous lectures, where it has a little bit of a, what they called an activation loop or an activation lip, that is the part that's going to be covered over until something happens that springs it into activation. So the activators can be generally such things as calcium or calmodulin in this particular case, or it could be often a phosphorylation mechanism or an interaction with some other kind of protein that will now spring this autoinhibition site open. So our calcineurin structure here for this is that it allows us to have a calmodulin binding domain, our autoinhibitory domain, and our catalytic domain. And our activators can be our calcium and our calmodulin, and we can also see that there's some binding sites for some other iron and zinc proteins. Now our calcineurin B subunit can be myristylated, and so the myristic fatty acid gets bound to it at one of the N-terminal glycine, and it will now be able to bind to the membrane when that happens. So that could actually anchor it to the membrane if it gets myristylated.
What does that mean?
There are proteins that would normally be globular and in the cytosol, but if they need to get to the membrane, then they get a protein that can put a palmitoyl group on or a myristyl group on, and that will then get it to the membrane. So you'll notice that when we had our beta-gamma subunits, those beta-gamma subunits had a lipid on it, it happened to be farnesyl, but it is going to allow that beta gamma to hang onto the membrane by the lipid group. So otherwise, it would be free floating in the cytosol, and it wouldn't be where it needs to be. So that gamma subunit always has this lipid on it. It gets the lipid put on it when it's made before it even goes to the plasma membrane. The alpha subunit also is lipidated, and it's either got a palmitoyl or a myristoyl group on it, or both. And that's what allows the beta gamma, or the alpha subunit, to stay in the membrane. So otherwise, they wouldn't work. So this one also. So if it's going to be myristylated, that tends to be actually, there isn't a way to remove that. If it's palmitoylated, there are enzymes in the cell that will take that palmitoyl off. So that can actually be a regulatory mechanism by which you can keep your proteins either in the membrane or out of the membrane. Okay, so our having zinc or iron is not an unusual situation. You've got lots of proteins that you know of already that have iron embedded in them. And I guess you also know that if you've got a cold, you can take a vitamin pill that has zinc in it. So that's not an unusual regulatory mechanism. So you can see that we're spread out here from N-terminal to C-terminal. Our catalytic domain occupies most of this A subunit, and then we've got the binding site for the B subunit here, a binding site for our calmodulin over here, and our autoinhibitory site. So you can see our site over here on the B subunit is where we're going to be binding some calciums, because you can see our B calcineurin subunit has 4 EF hands. So not only are we regulating by calmodulin, we've got this B subunit itself, which has the EF hands that can bind to calcium. So you can see it's very highly calcium regulated. So we have a low calcium in active form, and then as we get calcium binding, the calcium binds to these sites on the B subunit. That gets it partially activated, and then to top things off, our calmodulin can bind, and once that binds, now we have a complex that is fully activated. So you can see you've got different levels of the ability of this phosphatase to be able to remove phosphate groups. Now, how does this fit in here? So here again, we've got a dopamine receptor as an example, and this is a D1 receptor. It is going to be activating cyclic AMP production through its GS and its adenyl cyclase. This is a particular substrate for PKA, and it gets activated. And it turns out this is a regulatory protein that regulates phosphatases. But when we have calcineurin and a competing mechanism that brings in calcium, so the example here is the NMDA receptor. The calcium comes in response to the glutamate in a neuron like this. This allows this calcium now to activate the calcineurin. It does it not only through the B subunit, but it also does it through the calmodulin, and then this can remove those phosphates. So this is just one example of the many substrates that could be regulated by two competing mechanisms. A mechanism that phosphorylates it by PKA, and a mechanism that removes it by a calcium influx into the cell. Since this is a regulator of a protein phosphatase itself, it inhibits it. You can see that we might have some dual regulation further on. because if this gets inhibited and it can no longer dephosphorylate CREB, it would allow pKa to phosphorylate CREB, and you would get a much larger response than you did before. So our functions, with an emphasis on the brain, because there's more calcineurin in the brain than there are in many of the other tissues. We can see that it can be a regulator of transcription factors. We just looked at how it can be regulated through CREB, and the NMDA receptor is a good source of those calciums. The IP3 receptor inside the endoplasmic reticulum that allows the calcium mobilization from internal stores. These are all very good regulators of the calcineurin. We also see that in other areas of the body we have interleukins, interferons, cytokines that can be regulated. Our substrates, growth factors, heat shock proteins, tau, the NMDA receptor itself, are all likely substrates. In the rest of the body, there are many substrates in the thymus to regulate through interleukins. We've got regulation in the spleen. Spermatogenesis is regulated by our calcineurin. In the kidney tubules, diabetes with our synthesis of insulin. This particular phosphatase can impact many aspects of cellular regulation in addition to what's going on in the brain. So if we don't have calcineurin, and we can study this in knockout animals, we see that we have a dysregulation of our action potentials, dysregulation in our calcium signaling. We have effects that are occurring in the hippocampus, and it's been associated with schizophrenia. In other parts of the body, calcium regulation through this calcineurin can ultimately affect the way cardiac function is taking place. So we have diseases that the disruption of calcineurin can be involved in.
So have they actually seen, like, in schizophrenic patients, lower levels of calcineurin?
It doesn't have to be lower levels. It can just be mutations or SNPs. Okay, now let's switch over to a tyrosine phosphatase, and we've got PTP1B. There have been a couple that we've seen in our previous slide sets. SHIP is another one. They all work fairly similar to the way this PTP1B works, but they may have some subtle changes, like whether they're going to be mostly cytosolic or whether they're going to go to the membrane. I think we can use this as a prototype of this whole family. So here we have spread out again from the N-terminal to the C-terminal. We've got a fairly large catalytic domain, and we have initially what would be a endoplasmic reticulum targeting domain, because this is what really gets put onto the protein as it gets translated, but it does need to be removed, and it gets removed by a protease called calpain, which is under the regulation of calcium as well. So this signaling mechanism gets removed. And then you can see that we have lots of opportunities for regulation in this protein that are identified here. We can have it phosphorylated at serines, and here's serine 50. It gets phosphorylated by the AKT, or the protein kinase B. We also have in this proline-rich domain the proline-rich domain is going to be bringing in SH3 domain containing proteins. And so it can be one that binds to Rab, and it can, this little protein then can scaffold it to other kinds of proteins. And it can be phosphorylated here by bringing in SARC. So SARC was that non-receptor tyrosine kinase that's found in the cytosol. So these can come in, bind to these prolines in this proline-rich domain, and then we can get that interaction at this site. We can get our phosphorylation of our tyrosines, and we can get that through the insulin receptor substrate kinase, which is likely to be phosphorylating over here on these tyrosines. And then we can get some other changes. We can get oxidative changes on cysteines that are vulnerable, and there's one in here that can be oxidized. And we can also see something that we haven't talked about before called sumylation. Now, yesterday we talked about ubiquitination as a mechanism of allowing proteins, particularly those that have just been phosphorylated on tyrosines, to be brought to proteasomes for degradation. Well, there's a very similar kind of group called a sumoyl group, and that has sites on certain proteins that will do essentially the same kind of thing. So we've got some of those sites located here. Those are going to be lysines. So you can see we've got lots of regulation, and our regulation can be taking place as the cell the cell needs to have regulation of the protein for phosphorylation and dephosphorylation regulation. So here's a little bit about how it works. Here's our substrate over here, and our phosphotyrosine substrate in the green is going to be intercalating into the catalytic site, which is this activation loop over here, and then the phosphate will be able to interact with the aspartate and be hydrolyzed. It gets held into place by an interaction with another aromatic residue, this tyrosine, which helps to hold this in place by having aromatic stacking inside this protein. So we have our substrate recognition largely through here, and then our removal of our phosphate by these other sites. So our initial hydrolysis is going to be attacking this with water, removing the phosphate, and then removing the substrate. So this interacts with our insulin receptor substrate proteins that we saw when we looked at the growth factor receptors. It turns out that even though these were initially discovered to bind to the phosphotyrosines on the insulin receptor, this family of proteins does interact with other growth factor receptors as well. We've got our JAK, which is our Janus kinase, in response to leptin. This is going to interact and allow the phosphorylation on the tyrosines to signal. Our growth factors and our SARC. So how is this going to work? So we've got our activation of our kinases. Here's an example here going through our insulin receptor. our insulin receptor substrate binds. It can activate kinases, and once this gets phosphotyrosine on it so that it's activated, our protein tyrosine 1B can remove this and block this further activation in this pathway altogether. Here's our leptin receptor. Our leptin receptor through our JAK kinase is going to be phosphorylating a tyrosine, and our phosphotyrosine 1B can take this phosphotyrosine off and inactivate the JAK so that it no longer is going to be setting up this pathway. Now here's another protein phosphatase called SHIP, and this can work the same way. Here we are blocking a pathway going through the SARCs. So this SARC is going to be able to phosphorylate on tyrosines many substrates, but it needs to have a tyrosine phosphorylated itself, and so when this removes it, then our SARC is no longer active. So that's all I have to tell you today about phosphatases, unless you have some questions or some discussion points. We're done early. I guess that exonerates me from keeping you 10 minutes over yesterday. I would be happy to have all your homeworks over the weekend as you're doing them. And if you have any questions, I'll be happy to answer them. Okay, well, have a nice day.
Nobody.
What?
Is the new homework posted?
Yes, I emailed. Just in case.